would be if this was a much larger class. So let's try and make the best of it. So this class is going to be dedicated to organogenesis. So there are two classes. There's a class today and there is a class on Monday. And the way I would like to sort of structure this class these two classes is the following. So from my experience, having taken developmental biology as a graduate student, I mean, what I what I draw is the following. I mean, most classes in developmental biology tend to be very jargon filled. They tend to be very detail oriented, and so if you haven't had any background in developmental biology, these classes can be extremely difficult to follow and tend to be very concept poor, to be honest with you. At least this is what I recall. So my approach is very simply to lay a foundation or a framework for organogenesis, which is going to be very simple and conceptual. And the idea here is then that you can build on this framework when you listen to uh, a discussion or participate in a discussion on any aspect of organogenesis in any system. So what I will try to do in the process of discussing the organogenesis of the lung today is give you that framework for how to think about organogenesis in the broadest possible terms, uh, using the vertebrate, at least the mouse, as a model. So this leaves open the class for Monday. And so here, what my recommendation to you would be to sort of take a look at what we cover in the class today and then get a sense for what you think would be an interesting direction to go in. So there are two possibilities. The one possibility is, can we do a survey of the principles of organogenesis for another organ? I tend to be focused on endodermal organs like lung, pancreas, gut, and so on. So for example, could we do a class where we look at the organogenesis of the pancreas? And so then that will give you a broad overview of both lung and pancreas, and from it you can distill concepts that are relevant more broadly to organogenesis. The other possibility for the next class is that you look at whatever it is that we discuss in this class, and you say, no, look, this aspect of organogenesis seems particularly interesting conceptually. Why don't we go a little bit deeper in terms of understanding some of the nuts and bolts that go into shaping our understanding of that aspect of organogenesis? Okay, so my recommendation to you would be to sort of take a bird's eye view of the concepts that are discussed. They will be presented in the context of the lung. And then we can either repeat this exercise for another organ, or we can go deeper into some aspect of organogenesis. So depending on what the class decides at the end of this class, I will decide what we will cover in the next class. Again, my goal here is not to swamp you with information, not to swamp you with jargon. My goal here is to very simply create for you a framework for organogenesis that you can apply generally across all animals. Yeah, okay. So with that said, let's just dive right in. Uh, can you see my cursor, by the way?
Yes.
Yes.
Yeah.
Oh, good. Then I don't need the pointer. All right, excellent. All right, so this here is an overview of vertebrate development. So let's all get on the same page in terms of how, for example, a mouse or a human being is born, right? So it starts with fertilization and the zygote forms. The zygote then begins to divide. This happens very quickly in the first couple of days. And then 4-cell stage, an 8-cell stage, a modular, which is 16 cells and greater. And then this modular begins to implant, undergoes self-organization to create an extra embryonic layer, which is the trophoectoderm. And in addition to that, it creates this magical cluster of cells on the inside called the inner cell mass. This is the blastocyst. This is the first stage in embryogenesis where you see a clear segregation of tissue types, where you have an inner cell mass and you have an outer trophoectoderm. And I'm guessing you've covered all of these things in the past. And then this blastocyst implants in the mother. And post-implantation, the most miraculous thing happens, which is this loosely arranged cluster of cells called the inner cell mass forms an epiblast, which is a layer of epithelial cells. And then an even more remarkable thing happens in that in going from a hollow sphere, which is surrounded by an epiblast. Now remember, this is referring specifically to the cells of the inner cell mass. We're not talking about the cells of the extra embryonic tissues that are also developing at the same time. uh, the epiblast then undergoes a process that Lewis Wolpert at least thinks is the most important thing in your life, which is gastrulation, where you go from a single sheet of cells to three different layers of cells, an ectoderm, a mesoderm, and an endoderm. And once you form these three layers, of which the ectoderm and the endoderm are both epithelial in character, which means all of these cells maintain junctions, whereas the cells of the mesoderm, for example, are loosely organized. They may cluster together, but there are no defined cell-cell junctions among the cells that constitute this germ layer. So you've gone from the epiblast, which contains a single sheet, to three layers of cells: ectoderm, mesoderm, and endoderm. And then these three layers start doing some very interesting gymnastics to start folding and stretching, and they create basically an embryo which now contains two hollow tubes that run from anterior to posterior. One of these tubes, of course, is going to form the central nervous system, the brain, and the spinal cord. And the other tube that forms is the tube which is referred to as the gut tube. And from it, all of your visceral organs will arise. By visceral organs, of course, I am referring to the organs of the GI tract. And then in addition to that, you have the thyroid, you have the lung, you have the liver, you have the pancreas, and so on and so forth. And so, yes, so in going from this stage right here, which is a single layer, you've gone to three layers. Then the ectoderm and the endoderm have undergone tube morphogenesis to create two tubes that are running from head to tail. One of them will CNS, the other will form all of the endodermal derivatives. And of course, all of this is happening at a time when each of these tubes and the whole embryo is sort of undergoing these very complex gymnastics, as I mentioned earlier. But we don't need to worry about all of that. And then once these tubes are made, the process of deriving organs from these tubes and specializing cells within the organ start, which is really the domain of organogenesis. And finally, the animal is born with organs in some, with some resemblance to what these organs will look like in the adult, but are not fully formed yet. And one aspect of this entire process that is often ignored in most developmental biology textbooks and is really a black box in our understanding is what are the processes that happen once the animal is born that then allow the organ that is made, where all of the pieces are sort of in play, to then become a fully mature organ. This is really very poorly understood for most organs, and it's something that we won't be discussing in any greater detail. We will focus specifically on the events that happen in this domain, which is post-gastrulation leading to the formation of these organs. Right. So my emphasis is on endodermal organogenesis. In other words, the derivation of organs from the gut tube, which then are commonly referred to as all of the visceral organs. So basically, the thyroid, the lung, the liver, the pancreas, the stomach, and so on and so forth. So in the course of the lecture today, what we will do is very simply discuss uh, the derivation of each of these, the derivation of the lung in particular, but we will start from unpatterned gut tube through how the lung develops and how the cells of the lung become specified. And then finally end with some discussion about how are these principles relevant to how the lung maintains itself in the adult animal, right? So what you can see here are, of course, actually real tissue exhibits of the cartoon that you saw earlier. This, of course, is the epiblast. And from the epiblast, you then you basically fold the endoderm to make a gut tube that extends from head to tail. What you can see at the other end is the neural tube, which again runs from head to tail. And then from this gut tube, this embryo then folds in this direction to create an embryo that looks like this. And then what you can see outlined in yellow is really the gut tube which will then begin to specialize into different organs that are sort of shown here. So from this single hollow tube, we will then start undergoing branching to create other derivatives, which will then make each of these organs. So in the lecture today, or the discussion today, is going to focus on the organizing principles and the processes that are associated with this specialization. And my message here would be that the principles that apply to one of these organs can be more generally applied to the development of many other organs, not just invertebrates, but in other animals as well. Right. So the organ that we're going to be focusing on today is the lung. This is the organ that I'm interested in, mostly from the standpoint of my lab. And I want to start by sort of giving you what is a classic anatomical description of the lung. And the point here is to highlight two things, three things actually. The first is that the lung as an organ is a pretty complex entity in that it contains many different tissue types that are derived from many different germ layers. This is important. So, when you typically think of the lung, what you think about is the passages for air into and out of the body and a surface through which gas exchange occurs. This is generally the concept of what a lung is. But if you really think about it, for this organ to work, there are many other tissues that are required. For example, this branched network, which is responsible for the transport and exchange of gases, also has to be richly supplied by a vasculature, meaning a collection of blood vessels, which can then allow for the systemic blood supply to either be oxygenated or deoxygenated, whichever way you choose to look at it. In addition to that, what we also know about the lung is that in addition to this branched tree and a rich supply of blood vessels that connects with the tree, there are many other mesenchymal cell populations. For example, there are immune cells. There are muscles, smooth muscles in particular, which surround the branched network, of airway branched network that exists inside the lung, and so on. Now, the reason I'm pointing this out to you is because while I describe the organogenesis of the lung, what I'm really going to be focusing on for the next 5 to 10 slides is the organogenesis of this branched network that is responsible for the transport of gases. This part of the lung is what is endodermally derived. The rest of the lung, meaning, for example, the vasculature that associates with this branched epithelial network responsible for gas exchange, is mesodermally derived. In addition to all of this, you might imagine that breathing requires nervous input, right? So there are centers in the brain that control breathing, which means that the lung must be innervated, and indeed it is. So there are nerves in the spinal cord, there are nerves in the brain which send processes into the lung. There are also neurons that exist within the lung, meaning the cell bodies of these neurons exist in the lung, which are also locally modulating the activity of the lung. Why am I making this point? I'm making this point because, as you know, neurons and the brain in itself is ectodermally derived. So while the lung as an anatomical entity contains cells derived from multiple germ layers, for the point of today's discussion, we are going to discuss the organogenesis of the epithelium that is responsible for gas exchange within the lung, right? Okay, so that's the first point, that as an organ, it contains many different cell types that are derived from many different germ layers. But colloquially, when we say lung, what we are referring to is the epithelial branched organ that is responsible for gas exchange. So now the other thing which is important anatomically is that the lung is much like a tree in that there are branches and there are leaves. So the airways, if you will, are the branches, the trunk and the branches of the tree. The alveoli, if you will, are the leaves of the tree. So the airways are responsible for the transport of gases into and out of the lung. The alveoli constitute the surface across which gas exchange occurs. And so what you're seeing here, for example, is a representation of the airway, which is a highly branched structure, contains about 14 to 17 branches in the mouse and contain upwards of 30 branches in the context of 30 generations of branching in the context of the human lung. And then these branches finally converge on these little epithelial sacs called alveoli through which gas exchange occurs. These alveoli are very richly supplied with blood vessels. In fact, the interface of the alveolar epithelium and the capillaries that supply the alveolar epithelium is about 200 nanometers in thickness. So it's a very, very narrow interface, a very thin interface across which gas exchange effectively occurs. So yeah, so the principles here, or when you think of the lung as an organ, it contains many different cell types that are derived from multiple germ layers. Colloquially, when we refer to the lung, we refer to the branched gas exchange part of the organ. And yes, this part of the organ can be divided into two segments: the airways, which are responsible for the transport of gases, and the alveoli through where the gas exchange actually occurs. Right. So all of that is sort of summarized here. I won't repeat it. except for just pointing out to you that the epithelial cells of the airways and the alveoli are endodermally derived, meaning from the endoderm. The airway smooth muscle is mesodermally derived. The blood vessels and capillaries are also mesodermally derived. All of the immune cells that exist in the lung, which help the lung cope with infectious agents that are inhaled, for example, are also all mesodermally derived. And the nerves that are regulating the breathing activity of the lung are ectodermally derived. So yeah, just to set in mind what the entire organ is all about. Okay, so in today's class, we're going to discuss very briefly three things. How is the lung made? How does it repair itself? And we'll only go very briefly into this aspect of it. And what are the concepts that we can learn from how the lung is made that are applicable to other organs, right? So let's get started. Okay. So as I said, it's an endodermal derivative. So let's just quickly recapitulate what we know. Epiblast undergoes gastrulation. During the process of gastrulation, it is believed that there is a clear segregation of ectoderm from mesendoderm. There is a progenitor which can give rise to both mesoderm and endoderm. There is some evidence that this is indeed the case. And then this endodermal progenitor exits through the primitive streak and begins to make this endodermal sheet, which surrounds the ectodermal sheet. And this endodermal sheet then folds over to form a tube, and I'll show you how that happens in a little bit. And then that tube gets subdivided into the foregut, the midgut, and the hindgut. And then from these different segments, you derive different organs that make up all of your visceral organs in the body. Right. So how does the gut tube form? And this, what you're looking at here, is what happens in a mouse. I think it's important to appreciate that many of these gymnastics that occur during embryogenesis can be different from one vertebrate to another. But I think some of the organizing principles behind how these things happen, that these things happen and how these things happen, are conserved across animals. So in this particular case, what you can see is that there are folds that form in the anterior and the posterior end of the animal. And this fold then ultimately closes on itself to create a tube. What you're looking at here is a section. And then this entire thing sort of folds, and then this entire thing sort of folds onto itself to create here the tube that constitutes the central nervous system. What you can see here are the somites. These are, of course, mesodermally derived. And then, of course, next to the somites, what you have here is the gut tube that forms, right? So once gastrulation occurs, this is the sort of the spatial arrangement of the cells and how you basically go from just an embryo with 3 layers of cells to an embryo with a gut tube. So then starts the process of specifying this gut tube along the head-tail axis into different regions: the foregut, the midgut, and the hindgut. And generally speaking, all of development is literally about making boxes and then refining these boxes into smaller boxes and so on and so forth. So how do you pattern this gut tube into these 3 different components? Well, one of the most important signaling pathways that is relevant, 2 of the most important signaling pathways that are relevant here are Wnt and FGF4. There is also BMP. So there is basically a gradient of Wnt signaling. Where is this Wnt signaling coming from? Well, this Wnt signaling is coming from the mesodermal cells that are very closely opposed to the endoderm. Remember, gastrulation creates 3 layers. They remain opposed to each other all through development. And so the posterior mesoderm secretes very high levels of Wnt, FGF4, and BMP. And what this effectively does is begins to pattern the endodermal gut tube along the anterior posterior axis into 3 domains: the foregut, midgut, and hindgut, which can be distinguished by the expression of these different transcription factors. One other important thing to remember, so the concept here is that there is a fundamental epithelial to stromal interaction that begins to play a role in patterning this long tube along the AP axis. And this is driven largely by the expression of Wnt, FGF4, and BMP in the mesodermal cells in the posterior part of the animal. The other important thing to remember is that the order in which the cells are exiting from the primitive streak during gastrulation, endodermal cells in particular, also has some role in terms of how these cells respond to this gradient of Wnt and begin to turn on the expression of genes. So this patterning of the gut tube is driven by 2 processes: A, the epithelial stromal interaction through Wnt, FGF4, and BMP. Second, the order in which the cells are regressing through the primitive streak to create the endoderm. The cells that come out first are the cells that typically make the anterior, and the cells that come out later in the process of gastrulation are cells that typically make the posterior. So together with these epithelial stromal inductive interactions, there are also cell intrinsic processes that are built into these cells during gastrulation that then allows them to pattern into these three distinct domains. So here, what you can see is sort of a picture of this primitive gut tube that is in the process of being patterned. What you can see here, for example, is Wnt FGF4, which are acting at the posterior end here to repress anterior identity. And yes, together with that, what you can see here, of course, is the cardiac field. You also begin to see inductive interactions emerging from the mesoderm and the mesodermal derivatives that are in the anterior part of the animal, namely specifically from the cardiac field, which then begin to impact the epithelial cells of the gut tube and induce changes in gene expression, which are then beginning to pattern each of these organ primordia that are going to arise from the gut tube. And of course, this brings us squarely to the lung. I don't think all of these details concerning precisely what the signaling pathways are and how they regulate each other is so important. All that is important for you to understand is that the endoderm is a gut, is a tube. Around it, there are these mesodermal cells, and these mesodermal cells are actively going to produce signals like Wnt's, for example, and FGF10, which are then going to induce the gut to begin to form little protuberances, here the right and the left lungs. And then this structure, even though we draw it in this manner, this entire tube is sort of bathed or surrounded by mesodermal cells. And this is extremely important to understand because it's the interactions between the epithelium that constitutes this structure and the surrounding mesodermal cells that is effectively then going to drive all of these branching events that then give rise to the lung. So one tube, order in which you egress from the primitive streak, epithelial stromal interactions, principally the Wnt FGF4 BMP gradient from the posterior, act to divide this tube into three parts. Remember, the entire system is developing, and so the cells that are both in the endodermal, in the gut tube, and the mesodermal cells around them are also developing. In time, again, the mesodermal, the epithelial mesodermal or mesenchymal interactions will then begin to induce localized changes in gene expression. In this case, you'll begin to see the expression of a transcription factor called NKX2.1, which then marks the site from which these buds form. And then these buds, again, via a process of reciprocal epithelial stromal interactions, will undergo a series of branching morphogenesis. So in the next couple of slides, we're going to discuss what are the processes that drive branching morphogenesis. So, if you think of the lung as a tree, is the lung in one individual the same as the lung in another individual? Broadly speaking, the structure of the tree will be the same in that there will be branches and there will be, a stem or trunk, branches, and then there will be leaves. But to what extent is the structure of one person's lung similar to the structure of another person's lung? And how does this very elaborate branch network get made? What are the processes that drive this process, that drive this? So this was the subject of a very seminal study by a friend of mine called Ross Metzger. Ross spent about 10 years, actually, as a graduate student doing this work in Mark Krasnow's lab at Stanford. And what Ross did to try and begin to answer this question of how does the lung get made, and to what extent is the lung in one animal similar to the lung in another animal? And are there differences across strains, for example, or of animals? And so to try and address these questions in a meaningful way, what Ross decided to do was to take lungs from different stages. So just to sort of set lung development in the context of a timeline, the lung starts to form at about E9, E9.5 in a mouse, days post-fertilization. And then what you can see here are the sort of the stages of the lung at different days following the initiation of this event. So this is forming at embryonic day 9.5. And what you can begin to see here are Ross's staining of the epithelium of the lung bud with cadherin. And so what Ross did very systematically over this period of 10 years was to reconstruct each branch and its derivatives as a function of time. So in other words, what he did as a function of staining these lungs from different stages is followed how this branch, for example, begins to sprout other branches, and how those other branches in turn begin to sprout daughter branches, and so on and so forth. And what he was able to do by systematically following almost every branch in a developing mouse lung, and he did this not just for one strain, but he did this across strains, can be summarized in this beautiful diagram. What he showed basically is that the branching pattern in one individual is actually very similar to the branching pattern in another individual. And this branching pattern is created by three different subroutines of branching that are used in a very precise spatial and temporal manner. So let me just explain that. So what he showed is that there are three types of branching that happen if you consider the epithelial tube. So the one is domain branching, which is where you have a series of branches that form along one axis, and then the plane at which these branches form can change, and again you have a series of branches that emerge from that same parent branch to create a new set of branches. So if you were to look at them end on from a single branch by a process of domain branching, you could create a network of branches that emerges from it. The second type of branching event, or subroutine, which he described, is a subroutine called planar bifurcation, where the branches basically bifurcate, but these bifurcations are in the same plane. And so if you were to look at these branches end on, you would find that they are all in the same plane. So the last type of branching subroutine that he noticed is that the tips of these branches undergo a bifurcation, but they don't undergo a planar bifurcation, they undergo an orthogonal bifurcation. So if you were to look at these branches end on, you would see these four distinct, you would see the four endpoints aligned thus as opposed to thus. What was more remarkable is that because, in the process of carefully following each branch and the types of branching that were happening from each branch along each and every one of these lineages in the lung, is he noticed that the primary branching of the lung takes place via domain branching. It means when the lung initially begins to form, the subroutine for branching that you use is typically domain branching. And this has been color-coded beautifully in this panel here. Subsequently, what you see is that when you reach the extremities of the lung, you begin to see planar bifurcation, and finally, you see orthogonal bifurcation. So the large branches of the lung are created by domain branching, and as these branches become smaller and smaller and smaller, you see planar bifurcation and orthogonal bifurcation. The bronchoalveolar duct junctions, which is where the branched tree will end and the leaves or the alveoli will start, are largely generated by orthogonal bifurcation. So this was a remarkable study. And I think what is generalizable from this study is this idea that branching in branched networks, and almost every organ in the body has some branch network or the other, is largely going to consist of a stereotyped set of branches. Second, the stereotyped pattern of branches are going to be driven or generated by specific types of branching subroutines that are going to be used in a specific spatio-temporal sequence. Okay, so now comes back to the, let's come back to the question that we asked earlier, which is, okay, let's say that the branching in every one of us is more or less the same, but is it identical? I mean, is there any reason to suspect that one individual may have slightly different branches than another? And the answer to that question is yes. So what Ross found in this study of his is that across strains of mice, there can be stereotypical changes in branching such that certain lineages, for example, in the tree, may not exist in some strains of mice or do not exist in some strains of mice and exist in others. Overall, the branching pattern is conserved, but there are some branches that may or may not be present across strains of mice. Again, this is genetically hardwired in the sense that in that particular strain of mice, a specific lineage of branches will be absent. It is not like this is completely random. And finally, the question I think which will also pop up in your mind when you start thinking about how intricately this program is sort of laid out is, are there mistakes? Does the lung make mistakes in the process of branching? And the answer to that question is it does. In other words, what Ross found is that sometimes, so when you look at this diagram, for example, you'll see that there is a spatiotemporal sequence, right? So a particular branch will first undergo domain branching for one generation, maybe two generations, maybe three generations, and then the branches will switch from domain branching to planar bifurcation or orthogonal bifurcation. What he found in the context of the same strain of mice is that sometimes these branches forget. So what happens, in other words, are branches, for example, skip an entire subroutine. So there are examples where he found that there are some branches, and this was stochastic, meaning this wasn't stereotyped. Some branches would swap directly from a domain branching algorithm to an orthogonal bifurcation algorithm and completely omit planar bifurcation, for example. So, so these kinds of errors also occur. So yes, the answer to the question, is the branched pattern of the airways in the lung the same in every one of us? More or less. There can be some stereotype differences which can arise because of genetic background. There can also be some stochastic differences that arise because of errors that exist in this particular program of branching morphogenesis. So now the question,
Um,
sorry, yes,
I had a question.
yes, please.
So I was wondering, the stereotype differences in the lung. Sure, I suppose on a grander scheme of things, it would be quite robust as to the function of the lungs. But
Correct.
does it correlate to any like intricate differences when it in functioning when it comes to those stereotype
This is a
differences?
fantastic question, and there is no answer to this question. The reason is because at some level we are extremely limited in terms of how we assay lung function in the adult animal. So yes, I have asked Ross this question myself actually, and there is no answer. So I don't think we've developed the sophisticated kind of assays that allow us to, for example, compare across the strains of mice where specific lineages are deficient, to ask the question, is the overall process of gas exchange under normal conditions or under conditions of, say, exertion any different? We simply don't have that sophistication to ask that question in a mouse. And this speaks to a larger problem that those of us in the lung field have, which is the understanding that the lung is extremely limited in terms of what it can tell us about lung function. And this has sort of motivated many individuals now to, over the last 5 years, move to models like ferrets or pigs even, and created transgenics to this effect, which then allow us to ask the kind of question that you're asking in a more sophisticated model where we have more tools available to us. I don't know if that answers your question.
Yeah, yeah, yeah. Okay.
Yeah. So the answer is we don't know, but it's a very good question. Yeah. Okay. So
Hello.
yeah. Sorry. Yes.
So I have a question that is this branching continue throughout life or it's
Mhm.
restricted to the
Fantastic. Yeah.
embryo?
Exactly. Good. So that will yeah. So you just have to wait for about 3 slides, and I'll answer that question.
Okay, okay.
Is that okay? So remember,
Yeah.
you think about a tree. So we are gradually building a tree in some sense. So the first thing that generates the processes that I've described so far are the processes that give rise to the trunk of the tree and its branches. We haven't come to the leaves yet. And once I describe the entire process to you, then I don't have a specific slide that speaks to the question that you asked, but I'll tell you how we are to set this entire process in the context of the adult. Is that all right?
Yeah, okay.
Yeah. Okay. Good. Good. Yeah. Please stop me at any time. Yeah, because we tend to ramble and useless. Okay. Right. So I'm very sorry for this very simple picture, but I think it will convey the point that I'm trying to make, which is, all right, we've just looked at an extremely elaborate branching uh, program. And yes, this is going to be true for every organ, the liver, the pancreas, the gut itself. Every organ that you can imagine will probably have some branched structure, even the vasculature, for example. All right. So there's a stereotypical program that generates it. What are the mechanisms that drive this process? Well, again, we come back to a concept that we've already encountered, and that is the concept of a reciprocal epithelial stromal, or excuse me, epithelial mesenchymal interaction. So it turns out that there is one very, very simple signaling circuit that drives all of the branching events, all of the branching subroutines that we've discussed in the previous slide. And that circuit is very simply this. There is FGF10 production in the stromal cells, the mesenchymal cells that are very closely uh, associated with the developing, the branching epithelial tube. And these cells produce FGF10, which then acts on the cells of the distal tip of the branching tube to induce the expression of a gene called sonic hedgehog. And there is a reciprocal interaction between FGF10 and sonic hedgehog, such that sonic hedgehog represses the expression of FGF10. And this simple network, which involves, for example, the induction of a specific class of transcription factors in these sonic hedgehog expressing cells called ETV domain transcription factors, then leads to an iterative loop which drives all of the branching events. So in other words, once a branch forms, sonic is expressed, sonic represses FGF10, then by a more complex series of interactions, the FGF10 expression refines further. And then this branch can, for example, undergo a bifurcation event or undergo domain branching or undergo any other type of morphogenesis, right? So all of these branching events that we've looked at so far, doesn't matter whether it's a domain branching, planar bifurcation, or orthogonal bifurcation, is fundamentally being driven by this FGF10 sonic hedgehog circuit that exists at the distal tip of each of those branches. So one of the questions that you may, okay, before I move forward, I tried to look for this slide in the morning, but I couldn't find it. Please, if any of you is interested, please ask me and I will send the reference to you. So one of the questions that you could ask is, okay, great. It's FGF10 is being produced in these stromal cells that is then attracting the growth of the tube. And together with attracting the growth of the tube, it's inducing sonic hedgehog expression. And the sonic hedgehog that is expressed is then acting on the stromal cells that are making FGF10 to shut off FGF10. And so that shuts off this aspect of morphogenesis. Now new FGF10 expression comes on, and then the branch grows further. Excellent. But how does this explain the different branching subroutines that we've discussed? How does the epithelial tube that is branching know the difference between a domain branching or planar bifurcation or orthogonal bifurcation? And it turns out that there is no clear answer to this question, but there was a very interesting computational or modeling study that was done where people began to play with the dynamics of FGF10 and sonic hedgehog in terms of the intensities of signaling that are taking place, the time course of signaling that is taking place in this very localized milieu of the branch tip. And what they were able to show is that by simply altering the efficiencies of these signaling pathways, you could basically switch from a domain branching type branching subroutine to a more planar bifurcation or an orthogonal bifurcation type subroutine. I don't have a slide with the central findings of that study, but if any of you is interested in how this modeling was done to take this very simple circuit and apply it to a branching organ to show how you can generate different subroutines, let me know and I will give you the reference. So as I mentioned, and in the interest of time, you've now created a branched network. Yeah, these are basically going to make up the airways of the lung, the conducting airways, meaning the passages for air into and out of lung. The gas exchange of the lung does not happen here. The gas exchange of the lung happens in these sacs that culminate, that sort of form once the airway is made. And these sacs are structures called epithelium, are called alveoli. Now, what I'm trying to say, what is important here to understand is that the epithelial lining of the airways and the epithelial lining of the alveoli are continuous. So it's still one simple sheet, but it's a very different type of structure that forms. So in one case, you form this tree. In the other case, you now have to start forming these little saccules at the end of this branched tree. So how does this happen? Well, okay, so one very, just by way of
background, I mentioned to you, for example, that there is this tubular structure. This is the, what you're seeing here is the branch tip, for example. And yes, there is FGF10 that is being produced in these cells, and then this is acting on these
cells. Actually, there's an error here. I've reversed it. The green should be SOX9, the red should be SOX2. And so what you have here is a group of cells that begin to express this transcription factor called SOX9, and cells that are further away from the branch tip express this transcription factor called SOX2. And so what is happening in the context of the developing lung is that as the lung is growing, the distal tips of the lung are seeing high levels of FGF10. They're generating sonic hedgehog and all of that. And the structure is undergoing branching in one of various 3 subroutines. The cells at the distal tip of the lung express very high levels of this transcription factor called SOX9. And the cells that are further away from the distal tips that are not seeing very high levels of FGF10 are the cells that begin to express a transcription factor called SOX2. And this is important because as the lung is growing, as the lung is branching, what you're effectively doing is maintaining a population of SOX9 positive cells at the distal tips of this growing network and expanding the network of SOX2 positive cells further away. These SOX2 positive cells will be the cells that will then commit to making airway lineages. The SOX9 positive cells are cells that are multipotent in some sense, and these cells will proliferate to continue to give rise to this branching structure. And ultimately, these are the cells that will switch from generating SOX2 positive airway cells to generating alveolar cells. How does this happen? So it turns out that this entire process is actually driven not via local signaling, but in fact via a global signal that is generated during embryogenesis. So the processes by which this population here switches from making SOX9 to making SOX2, again, sorry for the label, this transition, this developmental program is switched off at some point during development by glucocorticoid signaling. So at the time when there is a glucocorticoid signal that peaks during embryogenesis, the leading edge, which is now expressing SOX9, ceases to now make SOX2 and continues to grow to generate a group of structures, a group of epithelial cells, which will then undergo alveolar morphogenesis. So in other words, just to be clear about it, if you were to, for example, stimulate glucocorticoid signaling earlier, then the extent to which the structure undergoes branching and generates airways will be smaller. And if you were to inhibit glucocorticoid signaling altogether, then you would never undergo the transition of cells at the distal tip from being committed to making airways to making alveoli. In other words, you would never generate alveoli at all. And this was the subject of a study in 2014. So at the time when the animal is born, what you've got is a branch network, the distal tips of which contain SOX9 that are continuing to grow, continuing to undergo some form of branching. But then there is a glucocorticoid pulse that limits the differentiation of these SOX9 positive cells into SOX2 positive cells that will make airways, so that the branched tips that exist at the distal tips of this tree now are now in a position to, again, through a series of epithelial stromal interactions, not make airway, but generate alveoli. Now, the question that was asked to me earlier was, does this entire process continue through adult life? The answer to that question is no, but there is definitely growth and there is definitely formation of some new alveoli that happens in the early postnatal period. I mean, as you can imagine, the size of your lungs when you're a kid and the size of your lungs when you're an adult are very different. And this, of course, involves the addition of cells to every compartment of the lung that exists at the time when you are born. But if your question is, does the process of branching morphogenesis continue through the adult? The answer to that question is no. By the time you reach adulthood, for example, that process of branching morphogenesis has almost completely stopped. And so what you're left with at that point is a tree and a set of leaves or alveoli, which are sort of set for life. At this point, any perturbations to this organization will require repair. There are some examples, controversial examples, that the lung can regenerate to some extent, but we can discuss that at a later time. I don't know if that answers the question.
Yes, it answers.
Okay, great. Okay, so now the question is, okay, so you've built a branch structure. This branch structure now sees glucocorticoid signaling, and so it continues branching to some extent, but then it stops. The cells that are the distal tip have stopped making airways and have switched to making alveoli. Now the question is, how do you begin to specify cells in the context of this branch structure? Meaning, how do you assign specific cellular identities? And here is where, for example, a very important signaling pathway that comes into the picture is a pathway that I worked on and continue to work on, which is Notch. And so what essentially happens is these are your SOX9 positive cells at the tip. These are all SOX2 positive cells that are further away from the tip. And what activates in these cells is Notch signaling, which then leads to the specification of secretory cells and ciliated cells and all of the other cells that comprise the airway tree. So if you just sort of try to imagine this process happening from say E11, so if we go back to that picture that I drew, this picture from Ross's paper. So the lung is undergoing branching. All of these tips contain SOX9 positive cells. At some point around this, you will begin to see SOX2 expression emerge on the parts of the tree which are no longer branching. Again, the overall domain of SOX9 will expand, as will the domain of SOX2. And at this point, you've got a whole bunch of SOX9 positive cells at the distal tip, and SOX2, for example, has now ceased or is going to cease expression along the major branches of the lung. And in the SOX2 domain, you're going to induce Notch signaling, which is then going to specify cells to make up the cells of the airways. So this is the mechanism by which you've gone from just a simple sac of cells surrounded loosely by some stromal cells to an intricately branched and now patterned organ. So this is a sort of a descriptive account of how lung development occurs. Let me just look at how we're doing on time. Okay. I want to now sort of give you a slightly different perspective on lung development. And what I want to do is to introduce the third concept. So there are two concepts that I don't know whether they've come through clearly, but there are two concepts that are critical to organogenesis. The one concept is the concept of a reciprocal epithelial stromal interaction. So there are signals moving back and forth between tissues that are regulating their development. The second concept is that these signals, as a function of going back and forth, are setting up gene regulatory networks in the context of each of these tissues, which is then allowing these cells to acquire fate. So the SOX9 is a gene regulatory network, SOX2 drives another gene regulatory network, and so on. So what I want to introduce now is the final concept in organogenesis, which is the concept of a stem cell. So the question that we're going to ask is, what is the relevance of the stem cell concept to organogenesis and how organs are made? So stated in other words, is there a singular stem cell type that gives rise to all of the cell types of the respiratory tree, meaning the airways and the alveoli? So I don't know whether anybody has been over this with you before. So in the interest of sounding really trivial, I will do this, which is to sort of just flesh out what is the concept of a stem cell. So a stem cell is operationally defined as a cell that can both make itself and can make other types of cells. Now, as you can imagine, during development, if you start off with the zygote, fertilized egg, this is a totipotent cell, meaning this cell can make every type of cell, both the embryo and the extraembryonic tissues that support it. Then as a function of time, this cell gradually loses potential. For example, it becomes a cell in the inner cell mass, which now does not have the capacity to make any of the extraembryonic tissues, but can make all of the tissues of the embryo, and so on and so forth. The point is that at every one of these stages, the cells that you define are cells that can both make themselves and make other types of cells. And so any cell that operationally fits this description is a stem cell. And so the question that we are asking in the context of, say, the branching morphogenesis of the lung is this concept at all relevant to the genesis of this organ? Can one say that the entire branched structure airways and alveoli originates from a singular cell type? This is the question. So this question can be experimentally addressed and has been experimentally addressed by looking at, for example, again, this idea of the gene regulatory network, by looking specifically for transcription factors that are expressed at the distal tips of a growing tree. So again, you'll recall that the lung is growing outward from the extremities, from the SOX9 positive structures. And so this is a study that was done by Emma. Emma's in Cambridge, Emma Rollins, and where she was looking, for example, for transcription factors that are expressed in the distal tips of a developing lung at E11.5 in earlier stages. And what she identified was a basic helix loop helix transcription factor called ID2. And what she noticed is that these ID2 positive cells are literally sitting at the tips of these branches that are actively responding to FGF10 and undergoing branching morphogenesis. She followed the expression of ID2 through lung development. And what she noticed is that the expression of ID2 in these distal tips is maintained all the way through E15.5, E16, when the process of airway specification and branching basically comes to a stop. So the next thing that Emma did to try and address this question is a technique that most developmental biologists need to be familiar with, which is lineage tracing. So what Emma did now is to take the ID2 locus and generate a knock-in mouse, which allowed her to express the Cre-ER T2 recombinase. So this recombinase is this is a Cre recombinase which can basically act on LOXP sites and recombine them out of the genome. This is a slightly more intricate version of that recombinase in that it is fused to an estrogen receptor, and so the Cre recombinase is largely cytosolic. Till one is to stimulate this fusion protein with an estrogen analog like tamoxifen, at which point the Cre-ER translocates into the nucleus to induce changes in gene expression. So by inserting the Cre-ER-T2 construct into the ID2 locus, what Emma was able to do was to drive the expression of the Cre-ER recombinase in these ID2 positive cells in a developing lung. And so the hypothesis that Emma had is that if the lung has a stem cell, it must be located, it must exist among these ID2 positive cells that are at the distal tips of the lung, which are fueling the growth of the lung. And if in fact, one of these ID2 positive cells is a stem cell that can give rise to the entire pulmonary tree, then I should be able to tag it irreversibly in an early mouse and show, in an early developing mouse lung, and show that the descendants of this tagged population of cells contributes to all of the lineages that exist in the pulmonary tree. And so the experiment that was done here is this ID2 ERT2 mouse was bred to a mouse that expressed a LAGZ under a ubiquitous promoter. Of course, the expression of the LAGZ in this mouse was contingent on an active Cre recombinase. And so the experiment that Emma did was to take these mice, which contained the ID2 Cre-ERT2, bred it to a ROSA LAGZ. ROSA is this ubiquitous promoter. And then she induced tamoxifen in these mice at early stages. And so what she found when she did this at early stages is, of course, only the cells at the distal tips of these lung buds were labeled with LAGZ. And then as she followed these lungs, so when she induced it at E13, slightly larger subset of cells, she induced them at E18.5, an even larger population of cells. But if she was to follow these cells that she labeled at early stages through subsequent stages of lung development, what she noticed is that the descendants of these ID2 positive cells then generated cells that were distributed throughout the airway and the alveolar tree. And when she phenotyped the cells, meaning she looked at whether these LAGZ positive cells expressed markers, what markers they expressed, what she found is that these LAGZ positive cells generated descendants that contributed to every lineage that exists in the airway tree. So I hope the experiment is clear. So the experiment was to take a group of cells that are sitting at the distal tips of the growing lung tree at an early stage of development and irreversibly label them with LAGZ, and then allow this lung to develop through to adulthood, and now ask which are the cell types that express LAGZ. And what they found is that all of the cell types in the lung express like Z. And so the conclusion that Emma came to from this experiment is that these ID2 positive cells that exist at the distal tips of the lung bud constitute a population of stem cells that account for the genesis of the entire airway tree. So is there a singular stem cell that gives rise to all the pulmonary epithelial cell types? The answer to that question is yes. Now, functionally, if you were to think about Emma's experiment, one other type of experiment that you might propose is it possible for you to ablate this cell? So can you take the ID2 positive cell and kill it by some mechanism, say by the expression of a diphtheria toxin? And can you show now that under these conditions, the lung is not made? Well, this is an experiment that Emma didn't do, but a colleague of mine at Boston University, Laertes, did this experiment, not with ID2, but with a different CRE-ER. And he showed that, in fact, yes, if you were to ablate these cells at the distal tips of a growing lung tree, that in fact, you wouldn't make a lung at all. So I think there is evidence now, both from the standpoint of lineage and from the standpoint of genetic evidence, that in fact, the distal tips of the growing tree, the SOX9 positive population, the ID2 positive population, you can give it many different names, do contain a population of cells that likely proliferate during lung development to both make themselves and to generate cells that generate all of the cells that comprise the respiratory tree. So the ablation experiments from the ID2 CRE-ERT2 have not been reported, but as I mentioned, a different set of ablation experiments have been done which validate the stem cell hypothesis. So with that, I'm going to summarize, I think, what is the take-home message from this class, and then towards the end, briefly talk about how all of this development at all relates to the maintenance of the lung in the adult, and of course, regenerative medicine. So there are three key concepts that drive all of organogenesis, I think, in terms of processes. One is reciprocal epithelial stromal interactions. It doesn't matter whether you go back to the earlier stages of gut tube patterning and you think in terms of Wnt, FGF4, BMP, specializing the anterior versus the posterior end of the gut tube. It doesn't matter if you're thinking in terms of FGF10 and sonic hedgehog acting reciprocally to drive the branching morphogenesis of the lung. All of organogenesis is driven through reciprocal epithelial stromal interactions. These epithelial or reciprocal epithelial stromal interactions drive and of course are driven by gene regulatory networks that are being established in the context of these cells. So in the context of branching morphogenesis, the FGF10 induces a gene regulatory network under the control of an ETS domain family of transcription factors, ETV4 or 5. And these gene regulatory networks then allow the stable pattern of gene expression to be established, which then drives the morphogenetic process. And finally, all of this is occurring, and all of these processes occurring in a context where there are indeed cells that have the capacity to make themselves and make other cells. And so these signals and these gene regulatory networks must at some level be maintaining these cells during the development of the organ, which then allows the entire organ to grow. And of course, these signals are being altered, which then allows cells that are multipotent to gradually become more differentiated and acquire character, and of course, complete the organogenesis of the organ. So, you know, this sort of a conceptualization and this sort of an articulation of development sort of begs one question. I mean, this is a question that I get asked every time when this class is sort of held. And the question that is asked is, you know, okay, so we know the signals, we know some of the components of gene regulatory networks. We know that there is a stem cell that is responsible for driving the genesis of the respiratory tract. Is there such a cell that exists in the adult animal? And is this cell the cell that is responsible for the repair of the adult animal? And the answer to that question is nobody knows. But what is important to understand is that it seems unlikely that that will be the case. So while we very naively imagine that the kind of embryonic stem cells that exist hello, am I still audible? Because there's been a power fluctuation actually.
Yes, yes.
Okay, sorry. Yeah, yeah. So, so naively, when we start thinking about development and we start thinking about stem cells and regenerative medicine, we imagine that there is this cell. Okay, so there is evidence that there is a cell in the embryo, which is a stem cell-like cell, which can make all of these cells. If you were to look in the adult animal, do you see cells of this kind existing in the adult animal? And are these the cells that are going to be repairing the lung every time it is damaged? And the answer to that question is no. So for example, where Emma did this study, so when Emma found ID2 as a marker for these distal progenitor cells in the developing lung, she looked at whether there were any ID2-like cells in the adult lung. Truth is, there is ID2 expression in the adult lung, but the ID2 expression in the adult lung is not in a undifferentiated population of cells, but in a differentiated population of cells. So ID2, for example, is expressed in the adult lung in the context of, say, alveolar type 2 cells, or in some airway epithelial cells. So the description of the stem cell in the fetal lung does not fit the description of any known cell in the adult lung. And the important concept here is that it is not sufficient to just simply follow a marker, meaning if you were to argue that, look, there is an ID2 positive cell in a developing lung, and it is by all measures, genetics, lineage, been demonstrated to be a stem cell, can one not argue that the ID2 positive cell in the adult lung is also such a stem cell? The answer to that question is no, because these patterns of gene expression change. And in fact, even though the gene may be expressed in the adult lung, there is no assurance, in fact, there is no evidence that it marks an undifferentiated population of stem cells in the adult lung. So yes, as far as our data currently is concerned, there is no cell in the adult lung that resembles that SOX9 positive, ID2 positive, multipotent progenitor that gives rise to the adult lung. So then the question becomes, okay, so that's a little bit disappointing because, we've studied all of this development and we've identified these stem cells, but, such stem cells don't exist in the adult lung. So what is the nature of the cells that contribute to the repair of the adult lung? And here is where I think the lung is a fascinating organ, because it turns out that, the different segments of the respiratory tract, the airways, the alveoli, they are maintained by different populations of cells. And what is most interesting here is that these cells that maintain these different segments of the respiratory tract are in fact cells that are differentiated, meaning they are cells that appear to be specified into cells of a specific type. For example, the cells that maintain the airways in the adult lung and repair the airways in the adult lung are airway secretory cells. Cells that maintain and repair the alveoli in the adult lung are alveolar type 2 cells. Both of these are cells that are apparently specialized cells that have the capacity, nevertheless, to make themselves and to make other types of cells. And so, this is the second concept that emerges from our study of lung development and homeostasis. The first is that there is a stem cell that exists in the fetal lung, but there is no indication that such a stem cell exists in the adult lung. And second, the kinds of cells that are maintaining and repairing the adult lung don't need to look like these naive stem cells that exist in the fetal lung. These cells could very easily look like differentiated cells and could be differentiated cells that exist in the adult lung that nevertheless have the capacity to make themselves and make other types of cells. And this is an extremely important concept, because I think this is a concept that applies to a large number of organs outside of the hematopoietic system. I think the concept that, the adult organ system or tissue system is dependent on a population of undifferentiated stem cells, just like in the fetal case, is arguably a highly specialized type or mode, which is seen in the context of the hematopoietic system, but not seen in the context of other organs or other tissues. Right. So one of the questions that clinicians often ask is this question. So if one is to imagine that there are cells in the adult tissue and that these are differentiated cells that can make other types of cells, what is the repertoire of cells that these adult stem cells can make? For example, can these adult stem cells that exist do these cells have the capacity to undergo germ layer transitions? So the big question, for example, is, can a cell in the lung, for example, which is endodermally derived, differentiate into a fibroblast, or differentiate into a blood vessel cell, or differentiate into a neuron? And the answer to this question is no. So all of these cells in the adult, which are nevertheless adult stem cells, don't have the capacity to cross germ layers and don't have the capacity to make multiple types of cell types. Why am I saying this? Because if you go back to the literature and look, you will find examples of papers, and this is a paper that was published in the New England Journal of Medicine, where people will argue that they found an organ stem cell. What does that mean? That this is a stem cell in the lung that can make not just the cells of the airways, but this is a cell that can make every other type of cell. For example, it can make blood vessels. And one has to be very skeptical about these kinds of claims because, to my knowledge, there are no cells in the adult animal which in vivo can undergo, can cross germ layers and generate cells of different types. So the reason for pointing all of this out to you is because I want to sort of distinguish developmental organogenesis in terms of the cellular basis for it from the processes that maintain and repair organs in the adult animal. The nature of the cells that do these two things are very different from one another in most cases. Of course, how are these cells regulated? Yes, it will, again, in the context of injury repair as well, also involve reciprocal epithelial stromal interactions and also involve the regulation of gene regulatory networks like you see during development. So I think that's a lot. So how are we doing on time? Yeah, I think what I will do now is stop. I have a little bit about regenerative medicine, but we can sort of go into that once I sort of get some feedback from you guys on what you think and how you plan to, how you would like to move forward. So as I said, the plan for the next class is to do one of two things. Either you guys can say, okay, I want to, for example, look at organogenesis in the pancreas. Or you could say that there are specific aspects of organogenesis that you've discussed that we would like to look at in slightly more detail in terms of signaling mechanisms, cell biology, and so on and so forth. So what is it that you would like to do for the next class? Let's sort that out and then we can move on to other discussions.
Arjun,
Yeah.
so you mentioned that in the hematopoietic system, there are these embryonic like stem cells which will give rise to lots of different cells.
Yes,
But in
even
most of the other,
yeah, but even in the context of the hematopoietic system, it's not so clear because blood cells in the fetus can have multiple origins, and the hematopoietic stem cells themselves in the context of the fetus move. So while there is a hematopoietic stem cell that gives rise to all of the lineages of the blood in the fetus, and such a cell or a cell of that description also exists in the bone marrow in the adult, it's not so linear, right? Because blood cells in the fetus and of course in the adult have multiple origins. I don't know whether this has been covered yet or not.
no.
Yeah.
So the thing
Yeah.
where I was heading towards was that in the case of lungs, you mentioned that these are not embryonic like stem cells, but adult stem cells, which not necessarily give rise to the whole organ, but then some subset of cells, and they do divide.
No,
so these are two contrasting
correct
stem cell types, right? Is there another one? Is what I was asking. Or does this cover the spectrum in adults?
Wait, okay. So important. Okay, let's go back to where we started. See, okay, so I mean, I'm sorry I keep repeating this, but I mean, I'm just trying to sort of give you guys a more holistic idea of how complex organogenesis is. So remember, I have just spoken. So go back to that picture where I sort of showed the lung in the beginning. I don't know if you were there, but so the lung contains many different things. It contains a branched epithelial tract, which is responsible for transport and exchange of gases. It contains vasculature. It contains muscle cells that associate with the airways. It contains a whole bunch of stromal cells, which include immune cells that are protecting the lung. And finally, we didn't even discuss this aspect of the lung. The entire lung is surrounded by a sheet, which is called a pleura, And this is extremely important because it gives the lung structural integrity. Now, why am I going into this level of detail? I'm going into this level of detail because, and of course it contains neurons, yes, of course. Yeah, it's because when you think about organogenesis, what you're really talking about is the growth of this whole thing, right? And the way we break it down is to start thinking in terms of tissue by tissue. So for example, E11, E12, which is when the first lung buds start to form and branch, is also the time when the vasculature in the lung starts to form. So the vascular tree, meaning the arteries, the veins, the capillaries, that must ultimately also form and integrate very intimately with the respiratory tract, also start to form then. Point here is that each and every one of these tissue compartments contains a distinct population of stem cells. What is important to remember is that every one of these tissue compartments is largely comprised of cells that are derived from different germ layers. The vasculature, for example, is derived entirely from the mesoderm. Immune cells, mesoderm. Muscle, mesoderm. Respiratory tract, endoderm. Neurons, ectoderm, right? So when you think about stem cells, even in the context, so fetus or in the adult, in an organ, always remember that these stem cells are going to be tissue specific and are largely going to obey germ layer identity, right? So even during development, once gastrulation happens, cells don't cross. So once you're committed to making ectoderm and mesoderm, endoderm, that's it. You won't cross that germ layer barrier anymore. So in the developing lung, you have all of these tissues from different germ layers, which have their own respective embryonic stem cells potentially, that will then generate each of these compartments. Now project into the adult, which comes back to the question that you asked. What is the identity of the cells in the adult? Well, there are two types, right? In the case of the hematopoietic organ, you could argue that there is a cell which resembles the fetal hematopoietic stem cell that persists into the adult and so on and so forth. But in the context of a solid organ like the lung, and if I look specifically at the respiratory tract, there are two kinds of cells. There are cells that have been specified, meaning they've committed to a specific fate and then acquired a specialized identity, which have the capacity nevertheless to differentiate and make other types of cells. There are some cells which seem less differentiated, more stem cell-like. For example, there is a population of cells in the lung called basal cells. These cells express transcription factors like P63 and so on. And these cells also contribute to the maintenance and repair. But again, so P63 is an interesting marker because, for example, if you looked at the skin, you would find that there are P63 positive cells that are necessary for the growth of the skin. There are P63 positive cells that form, that are the stem cells that maintain and repair the adult skin. But please remember, even in the context of the skin, the P63 positive cells that are giving rise to the skin and the P63 positive cells that constitute the stem cells that maintain and repair the skin express P63, but they are not derived from the same cell population. So the stem cells that, for example, maintain and repair the skin is a P63 positive population that is derived or differentiates much later in skin development than the P63 positive population that generates the skin to begin with. I mean, these are things that I'm sure others will cover and so on. But I don't know if I've sort of given you a flavor for what kind of stem cells exist in the adult. But it's a little bit more complex than we like to believe. Yeah.
Okay. Yeah. Thank you.
Yeah.
Hello.
Yeah.
I have a question that the pleural cavity, is this pleural cavity formed before the branching start in the embryo?
The pleura starts to form around the same time. Actually, E14.5, I think, is when the mesothelium starts to form. Give me a sec. Give me a sec. Bridget Hogan. So, okay, I can get back to you. There's a PNAS paper that describes that. It's a it involves a WT1 cascade. I can get back to you. Yes, around the same time.
Okay.
Around the same time. In fact, it starts, I think, around E12, E13.5. The reason I'm saying that is because there is input that comes from the pleura, which also regulates FGF expression that directs lung branching. So I think FGF7 and FGF9 are the 2 points of intersection between the FGF cascade that drives branching morphogenesis and the development of the pleura. But yes, I can send you that reference. Just send me an email and I'll send you the reference that will give you the precise time course.
Okay. Okay.
So, Arjun, a lot of times we hear that the damage to the lungs which happens is at times reversible in some cases and irreversible in some cases. So is it because that we have certain specialized cells which can particularly do some type of repair, and some degree of repair in some other cell types in the lungs is cannot be done? So is there some difference between that?
Yeah. So what is reversible and what is irreversible are, I mean, this is
Like in infectious diseases, some of the cases we hear that there is septic happening in the lungs, which is why the cells get damaged, and then there is some sort of recovery which happens. Like in COVID-19, we are also hearing that some patients are undergoing severe lung damage, but after weeks of infection, they start undergoing recovery, and after weeks they have the recovered lungs again. So is there some specific cell type which can be recovered and some cell types which cannot be recovered?
Okay. Yeah. Um, I mean, yeah, I think the question is extremely broad. So, all right, let's lay down some ground rules here. Okay. I think development and homeostasis are two very different milieus. Yeah. For example, development is in invertebrates, in mammals in particular, in utero. Yeah. Once you are born and you come into contact with the outside world, everything changes. The most important thing that changes is that you now have to contend with inflammatory and immune responses, which is how your body responds to the outside world, in addition to other physiologic parameters, of course. The reason I mention this is because the immune and inflammatory responses that occur in the adult animal are a bit of a double-edged sword. And this is particularly critical, for example, in the context of COVID-19. Why do I say double-edged sword? I say double-edged sword because while the response is adaptive in the sense that it allows the system to cope with whatever irritant or pathogen that it has come into contact with, you have to understand that this mechanism in itself can cause damage to the organ in which it is being induced. And this, for example, is the basis of this concept called the cytokine storm that has been debilitating to many COVID-19 patients. So in many patients, the problem is not with whether the organ has the capacity to repair itself or not. The problem is simply the fact that the nature of the immune and inflammatory response in these patients is so severe that the body effectively has no capacity to cope with it. So what you have induced is a downward spiral by which the body has lost all capacity to defend itself. So this is important because it allows you to tease apart why, for example, a person may not recover from why a person does recover. So if you don't control the nature of your immune and inflammatory reactions, you will ultimately suffer from whole organ failure, simply because these reactions unto themselves are toxic to the body, unless controlled very precisely. I don't know whether this concept is clear to you. So when you say, for example, that a person has recovered versus a person has not recovered, it could in part be because of the explanation that you provide, which is that in this one person, say a population of stem cells was more active than the other. But the more likely explanation, and specifically in the context of COVID, is that the person who's recovered was able to contain and control an immune and inflammatory reaction, which The person who did not recover was unable to contain. But let's get back to the idea. Your idea is, can you account for differences in outcomes based on the activities of stem cells? This is the concept. The answer to this question is absolutely. So if you take a disease like idiopathic pulmonary fibrosis, the reason it's called idiopathic is because people don't really know why it happens. Now, what happens in idiopathic pulmonary fibrosis is systemic scarring of the alveolar domain. Scarring is very much like the scarring that happens when you cut yourself. When you cut yourself, you form a scar. Similarly, a cut can also happen internally. It can happen because of, say, mechanical push and pull. It can happen because of an uncontrolled immune or inflammatory reaction. So there are scars that are, or there is damage and there are scars forming everywhere. So what happens in idiopathic pulmonary fibrosis is a process of wounding of the alveolar domain coupled with scar formation such that ultimately the lung loses all capacity to sort of expand, contract, and exchange air in any meaningful way. Now, even though the disease is idiopathic, there are experimental animal models that speak to what may be accounting for this scarring, this inability of the lung to repair itself in a meaningful way. It turns out that telomerase function is extremely critical. So if you, for example, remove telomerase activity, and this is a painful thing to do in the mouse because they have very, very long telomeres, but there are people, for example, who've made mice where they've basically over generations bred mice so that the stem cells in the adult lung have extremely short telomeres. And what they found is that in these mice, there is a predisposition towards alveolar scarring, fibrosis, and loss of lung function. So what does that mean in the context of the question that you asked? What it means is that in cells in which telomerase function has been perturbed, the cells in which telomerase function has been perturbed are less competent to divide and to generate other types of cells. So yes, in a patient in which you've completely ablated telomerase function, a person such as that is going to be more susceptible to alveolar wounding and scar formation. Scar formation essentially means there's no repair. The cells that have been lost have not been replaced. Than a patient in which, for example, a telomerase function is normal. I don't know whether that answers your question.
Yeah, it does.
Yeah.
Thank you.
Okay, so the question for you guys, there's a chat message. Oh yeah. Okay, so the question for you guys is, what do you want to do in the next class? Do you want to go deeper into some aspect of organogenesis, or do you want to do a broader survey of some other organ?
I think we can go into like a deeper aspect of organogenesis.
Like what?
That would be my preference.
Yeah.
Fine. Like what?
Such as I would suggest something like ECM, because I was expecting, like, because there is so much of morphogenesis happening at such an early stage, so you would expect a lot of ECM remodeling.
Yes. So are you Utkarsh, as in Utkarsh from
Yes,
Shikala's
yes.
lab?
Yeah.
Are you from Shikala's lab? No.
Yeah.
Oh, okay. Okay. So Utkarsh, let me ask you this question. I think it's an interesting question that you asked. But is there ECM, for example, when the lung is first forming, or when the skin primordium starts to form? When, for example, during development, do you have a fully formed extracellular matrix?
Yeah, I would expect it to be like at rather later.
Exactly.
Yes.
Exactly. So for example, it doesn't matter whether you look at the lung or if you look at organogenesis, even in insects. It turns out that most of this branching morphogenesis that I've just described to you is taking place at a time where there is no organized extracellular matrix. So genes, for example, collagen genes, I mean, so there are 4 core ECM components, right? I mean, laminin, nitrogen, perlecan, collagen, right? These are the 4. Now, it turns out that if you look at the time course of expression of many of these components are sort of dynamically changing during the development of the structure.
Yeah.
So I don't know to what extent, I mean, I guess the question that I'm putting to you is, talking about ECM in the context of development is a very complex problem, because there are ECM components that begin to be expressed during organogenesis, which most likely will affect some aspect of organogenesis. And then there is an organized ECM, which is what you typically associate with an animal once it is born, where you have a structured basal lamina, et cetera, et cetera. But none of this exists during development. So when you think in terms of ECM, what precisely are you talking about?
In the sense, like so like something which we are seeing is that, like, let's say these immune cells which get recruited in the skin, so they seem to be, like organizing the ECM in the sense that they are synthesizing new ECM molecules and or breaking down the few existing ones, and that in some sense aids in hair follicle development. So do you think like something synonymous occurring in lung morphogenesis and development as such?
It could. I mean, yeah, I think the more here again, the extracellular matrix is particularly complicated because it consists of a whole family of proteins that can have function independent of whether they are organized into a form that we typically recognize as an ECM in the adult. So, okay, I will keep this in mind when I look back at the classical models for organogenesis to see whether how ECM begins to plug into this process. But I'm not sure that there are any deep conceptual ideas that have been formed, particularly because I think the whole idea of what constitutes ECM during embryogenesis and development is itself so fluid. It is not the same thing as saying collagen is expressed or laminin is expressed, if you know what I mean. Yeah. So yes, I'll keep that in mind. If there are any other questions regarding what aspects of organogenesis you would like to go deeper into, please let me know, because then I will organize my presentation for Monday accordingly.
Arjun?
Yeah.
Arjun, some of us have a neurobio class from 11.
Okay.
So can we?
So can you please communicate your thoughts, whatever you have, to Varsha, and then she will get back to me with respect to what we should do in the next class. I won't hold you guys any longer. Bye. Yeah. Bye.
Okay.
Thank you.
Okay.
Bye.
Thank you. Bye.
Thank you, Arjun.